Thank you so much, Ahmed, for that introduction. And thank you for inviting me to the end of the year summit. I'm really excited to be here today to share some of the most important highlights of the safety initiative that New Harvest led with Vireo last year. First of all, for people that are not familiar with New Harvest, let me tell you a bit about our organization. New Harvest is a donor-funded nonprofit with the ultimate goal of guiding and developing the emerging technology and industry of cellular agriculture towards having the best possible impact on the world. Over the last decade, we've seen CellAg matured from what I would consider a sci-fi idea to an industry with over 80 companies, raising roughly 1 billion in VC investment. This made us realize that growth, particularly from industry, is well on its way. That is why last year, New Harvest changed its mission from building the field of cellular agriculture to maximizing the positive impact of CellAg. The new mission New Harvest has reflects our adaptive thinking based on the needs of the field. We believe that positive impact is not innately built into the advancement of a new technology. Cellular agriculture can spearhead change, but how it is developed, governed, owned, and implemented in society will dictate the impact CellAg actually will have on the world. For us, it's all about ensuring that CellAg delivers on its promises to make the world a better place. However, this will require a different type of experimentation that goes beyond the discoveries in the lab. It will require thinking around multi-stakeholder engagement models, self-governance models, potentially different approaches to public debate, and open data collection. Experimenting is something that we excel at New Harvest. And our first attempt was the safety initiative, which is the reason I'm here today. As we all know, the regulatory pathway for cultured meat and seafood is a top concern around the world. We know that regulations are country-specific, but the concept of safety is universal, and it is a vital requirement for cell culture products to enter the market. So, in order to advance safety and regulatory acceptance to enhance market adoption, there was an identified need to understand the current state of knowledge regarding the safety of cell culture meat products. Knowing that the promise of cell culture meat will never be realized if the products are not safe, the Culture Meat Safety Initiative was New Harvest's first industry-wide project designed to address a major gap in pre-competitive research, which is the lack of public data to help demonstrate the safety of cell culture meat and seafood products. This initiative, as I was telling you before, was led by New Harvest in collaboration with Vireo Advisors. It convened over 87 individuals representing 50 companies from around the world. In a series of workshops and one-on-one engagement, this group generated a collective understanding of the manufacturing process and identified safety questions that must be addressed. The findings have been shared on a co-authored peer-reviewed paper that has been recently published in the Journal of Comprehensive Reviews in Food Science and Food Safety. Today, what I intend to do is share some of the highlights of this initiative. So let's start. First of all, we raised the bar on food safety. Before this project, there was no public literature that focuses specifically on safety of cultured meat products. Our peer-reviewed paper was the first publication to address this major gap in the scientific literature. The way this paper was written followed a non-traditional approach to scientific publications because we didn't work with a university, and due to the novelty of the field, there wasn't a large body of literature to support the data collected. However, the paper has made great strides and has become an invaluable resource for advancing safety work in diverse areas, such as academia, workforce training, and education. And I'll talk more about that later on. So we intend for this paper to be a starting point for scientists around the world to generate a body of public data about the safety of cell culture meat and seafood. The second highlight was that for the first time, a generalized process diagram was co-created and refined by companies that shared previously unpublished details about their manufacturing processes. The objective of this diagram was to get a sense of the cell culture manufacturing process that is accurate and reflects reality, but at the same time, that is not overly academic, since we wanted this diagram to inform other stakeholders besides scientists. The diagram was developed using a descriptive approach rather than a prescriptive one, so that it could be applied to a range of manufacturing processes across the industry, sorry. That is why the structure, as you can see on the image, is modular, and manufacturers can choose which steps to use and move the pieces around to reflect their own process. The third highlight is that potential hazards were identified. Cell culture meat and seafood production may rely on processes that haven't been used for food production before. Therefore, it was necessary to assess the inputs, byproducts, and outputs of each manufacturing step to identify potential hazards. As we can see in this diagram, each specific process step was linked to manufacturing process hazard considerations that allow for further potential hazard identification. The table below the diagram shows an example. The source animal could introduce potential hazards such as bacteria, viruses, parasites, prions, and antibiotics. Therefore, it is important to understand the relationship between health status of animal cell donors and the potential to bring biological hazards into the cell culture process. Something to note is that some hazard considerations may be present throughout the process, whereas others are specific to only one step. Once we knew more about potential hazards, it was important to know what are the tools that already exist for simply and effectively managing these hazards and also assessing safety. The safety of these products is highly dependent on manufacturing processes that have product safety in mind since the beginning. Established practices and protocols from related fields could be transferred to the cell culture context. For example, hazard management practices such as good manufacturing practices from fields like food, feed, meat and seafood, pharmaceutical and medical could be applied to cell culture production processes. Safety assessment methods that exist may be also applicable, although some knowledge gaps were identified that create uncertainties with regards to safety assessment. The last element that allowed us to raise the bar of food safety was the identification of future research priorities. As I just explained, there are frameworks for risk management and testing methods used in other disciplines that can be applied to cell culture meat processes and final products. However, there are novel aspects that still need more understanding. During this initiative, we were able to collectively identify gaps that need to be addressed through research. Many of these priorities will require more collaboration between academia, nonprofits, government, and industry to ensure that research and the outcomes of that research are realistic. 19 research priorities were highlighted during this initiative, but as part of this process, participants voted for their top ones. 7 out of those 19 were voted as the most relevant. So I'm sharing the list of those of the top most voted ones on this slide. Finally, the safety initiative not only raised the bar on safety itself, but also on collective action. This initiative united the field around a shared understanding about the safety needs for cell culture meat and seafood. And this project was a success thanks to the collective effort. We were amazed by the generosity, openness, and participation of all the individuals and companies that joined. And I want to give a big shout out if any of you are in this call. We collectively identified what data is needed to demonstrate the safety of cell culture products. If we think about it, this would have been a challenging job for academics alone. So really, it was a participating companies coming together, which made this such a novel approach. The initiative also have a novel way at sharing information, because no NDAs were signed in the process of collecting data for the paper. This specific point was crucial because, as we know, cellular agriculture lacks public funding, so most research is conducted by VC-funded companies with strong intellectual property protections. To write this paper, we had to create new ways for companies to transcend individual interest regarding IP in order to create a body of public literature around safety. So we use our unique position as an independent nonprofit to gather credible data from leaders in the field through those one-on-one conversations and a series of workshops. Together, we co-created a piece of literature that is invaluable for continuing building the cell ag ecosystem, and that can help us minimize the risk of cell ag as an emerging technology that are associated with food safety. So what's next? One of our goals with the safety initiative was to create a starting place for collaborations around safety. And we really want to keep this momentum going. We will continue moving forward collaborative projects such as the one with Northeastern University's Biopharmaceutical Analysis Training Lab, or BATTLE, which is focused on developing an in-person safety training course. We are also developing an open source online course that we will serve as an educational tool for consumers, students, and regulators around the world who want to learn about the safety of cell culture meat and seafood. One of the priorities voted by participants was the development of standards. As the field continues to evolve, we will learn more about the types and concentrations of potential hazardous inputs throughout the production process. We will need to know how to measure and what elements are needed, such as the identification of maximum levels of residues, byproducts, or contaminants that will enable more efficient and reliable product testing. This initiative opened the door to more innovation in safety demonstration for cell culture products. We want to continue encouraging more open conversations with emerging leaders around the world about safety to generate the public data that is still missing and that was identified during this initiative. What we ultimately want to see is establishment of a body of public knowledge and talent that will allow the advancement of safe products to market for consumers around the world. So with that, I want to thank you for listening, and I leave my contact information in case you are interested in discussing more about food safety. Thank you.